Got it. Okay, so thank you very much for having me here today. So I'm gonna give you, it was a pleasure to come back because when I just started, I gave you a seminar talking about what I have been doing during my postdoc. It was really the first months here at Dalhousie. And now I would like to update you on what we have been doing in the past 4 years and a half. Actually, probably it was 4 years ago that I was here. And okay, so we still work in peroxisome, and we are interested. Oh, it's not moving. Oh no, it's moving there. It's not just the same written on my screen. Okay, so we're interested in peroxisome in the context of the immunometabolism. And when we talk about immunometabolism, we refer to how a canonical metabolic pathway can influence the behavior, development, and signaling of immune cells, both of innate and adaptive immune system, and also how immuno and inflammatory signal can actually alter the physiological metabolic status of an organism. And this branch of immunity was firstly theorized in 1883 by Ellen Netchinkoff, which kind of is a genius. He found phagocytosis, he theorized immunometabolism, and he also theorized the importance of the commensal for the host health. However, his observations stay unused for over 100 years, when then like around 50 years ago, some observations started, specifically studying obese mice, which found that in the adipose tissue of this mouse, there was a high and abnormal infiltration of macrophages. And then these studies were followed up, and they developed the concept that maybe metabolic dysfunction like insulin resistance in diabetes can be the cause or the consequence of this immunocell attraction and installment into the adipose tissue. And so as the year progressed since the 880s, so the publication number about immunometabolism increased, and they were mainly focusing in understanding how these macrophages in the adipose tissue of obese mice, so mice that were affected by insulin resistance, could actually generate a high inflammation in the host, and through some signaling were defined, like the gene kinases, the NF-κB mediated signaling that proved that the inflammation, the disease progression was caused by activation of immune cell as a consequence to a metabolic change in the host. And therefore, this led then around 2015 also to the development of treatment with anti-inflammatory to help people that have insulin resistance and other metabolic inflammatory disease. So what do we know today? Well, this is from a review extracted from a review of Luke O'Neill, which is at the Trinity College in Dublin, and is kind of like the immunometabolic specialist at the time of our age. And so through his career with other colleagues, he established how canonical metabolic pathway, the one you remember to study during your biochemistry 101 course, they all have also a function not only for energy homeostasis, but also to change in immunocells their behavior. For example, it is known that glycolysis is associated to pro-inflammatory phenotypes in macrophages and in T cells. Also, we know that fatty acid synthesis can promote pro-inflammatory phenotypes in what are called MA1 macrophages and T cells. And what a beta oxidation, so the degradation of fatty acid, as an shifted the cell to an anti-inflammatory phenotype. It is also known that amino acid metabolism are involved in the regulation of immunocell behavior, specifically arginine, glutamine, histidine. They all feed in the tricarboxylic acid cycle, and they promote a pro-inflammatory or anti-inflammatory phenotypes through the production of intermediate of the cycle. And so if we look at the cell, we see that all the major housekeeping metabolic pathway now have been linked to, they have been associated, they have been renamed that also as regulator of the immunocells behavior activation, and we will talk about also development. However, this diagram that again is extracted from O'Neill's review, it was published in 2016 and is already outdated because many other metabolic pathway or organelles that are contributing to activation of the immunocells have not been included just because they were not studied yet. And we are actually interested in some of the pathway that are not in this diagram, and specifically to the pathway that are linked to an organelle that is called the peroxisome. And the peroxisome are organelles present virtually in every eukaryotic cell. And as you can see them in these green dots in these figures, where the green is a scale antibody, which is a canonic marker to identify peroxisome in the cell. And so these organelles are essential for organism survival. And also they've been better characterized recently. They are very young. They were just discovered in the 60s. And we know that they are involved in the synthesis and breakdown of complex lipids and reactive anionic species. And they are also very important to produce important mediator of immunocell regulation, such as prostaglandins and leukotrienes. So in the past 10 to 50 years, experimental evidence accumulated, and they have demonstrated the peroxisome in specific immunocells help to carry out determine distinct immunofunction. For example, we know that they are involved in promoting phagocytosis by macrophages in Drosophila as in the murine system. They have been linked to the activation of MAB-mediated immunity as well as NF-κB-mediated immunity. And they have also been linked to some immunocell development processes such as T-cell differentiation, iNKT maturation, and potentially macrophage differentiation too, as I will show you today. So in my lab, when I started, we had determined, we knew something about peroxisome in immunocells, specifically macrophages, but we didn't really have an idea of the specific mechanism that peroxisome can be involved in regulating this immunocell behavior. So the overarching goal we set, it was to define peroxisome distinct signaling pathway that regulate inflammation, immunocell function, and development differentiation of immunocells. And we use the fruitful help, we take advantage of the fruitful help of the fruit fly, Drosophila melanogaster, because we are mainly interested in innate immunosignaling, and innate immunosignaling are highly conserved between invertebrates and vertebrates, because invertebrates have a specialized cell, such as macrophages, that can detect, engulf, and destroy a pathogen, and they can also secrete inflammatory and immunomediators, such as antimicrobial peptides and cytokines. So because we use Drosophila, because we had established so genetically amenable peroxisome biology is conserved from fly to humans, and also we have established some tools that allow us to specifically eliminate peroxisome in the cell tissue of interest. And we use the Gulf4-US system and the CRISPR-Cas9 technology to generate macrophages or fly with macrophages with no functional peroxisome. And to do that, it is sufficient in Drosophila to knock down one of the genes I'm here showing in this diagram, that are all the genes encoded the protein in this diagram, because these protein like Pexf, Pex1, or Pex2 are all involved in the import of the peroxisome enzyme into the organelle matrix during the biogenesis process. And therefore, if you eliminate any of these enzymes, you are blocking peroxisome biogenesis, and you have a cell that cannot do the metabolic activity of peroxisome. So as I mentioned, I had some evidence from my postdoc that peroxisome are important in macrophages. And here I'm showing you two of the key data. First of all, in vivo organisms, so in Drosophila, I generated this Drosophila with no functional macrophages in the no functional peroxisome in the circulating macrophages. And then I performed thoracic injection with a gram-negative bacteria, ECC15, which can trigger an immune response in flies. And we found that while these organisms is normally not lethal in the controlled fly over infection, when fly don't have functional peroxisome in the macrophages, they are highly sensitive to the infection and they die. A cellular analysis of these macrophages in primary cells and in culture cells show us that while control macrophages, when they are in contact with the pathogenic bacteria, can efficiently engulf them to eliminate the infection, then the peroxisome-depleted macrophages, they accumulate the bacteria on their surface and they cannot metabolize the bacteria. So we started to try to understand more why these fly are sensitive to the infection and how peroxisome can regulate macrophages behavior. We performed a multi-omic study where we did a transcriptomic and lipidomic on Drosophila macrophages like cell line, also known as SQ cells, and we treated this cell with a mock infection or with the ECC15, and then and we did this treatment in control and in peroxisome depleted or ablated cell, and then we collected after 4 hours upon infection the sample for global lipid metabolite analysis and transcriptomic profile, and performed an integration of the data. So we found in our lipidomic analysis that over 800 lipid species were changing up or down during an infection in macrophages peroxisome or without peroxisome, and we found a lot of differential changes. However, for today's talk and also for our study, we had to focus on something. And so for today's talk, I want you to focus on the phosphatidic that, as you can see, is a very low in concentration in Pexf depleted the macrophages during infection compared to the control, and doesn't really change that much in the cell of the two different genotypes if they are not infected, because this was the main principle of our screen. We were interested mainly in changes that happen during an infection, but not normally. Otherwise, we will be picking metabolic signaling, not immunometabolic signaling. And then for today's talk, I also wanted you to remember this difference. The PA, that is a precursor of PI, PS, it's instead very accumulated in the Pexf depleted cell during infection. And likewise is the DAG, which is also a precursor for PA and for PI and PS. So precursor accumulate in macrophage immunofunction peroxisome during infection, product are reduced. So we want to, of course, to also track some of these key lipids because we wanted to see whether it was just a change in the concentration or it was also a localization problem. So we generated a reporter to track this major lipid. And in this picture, I'm showing you a phosphatidic acid report, which we tracked in control cell and in Pexf depleted cell before and after infection. And what we found is a control cell before or after infection do not really show a lot of membrane or edge accumulation of the phosphatidic acid. However, the Pexf1 mutant, they have like some accumulation of phosphatidic acid in the membrane in mock infected cell, and then all the phosphatidic acid move to the membrane upon infection, confirming that there is an increase, and specifically in the plasma membrane, of these lipids, as suggested by the lipidomic analysis. We also tracked the PI reporter. Let's see how I can activate this video now. I think I have to shift. Oh, okay. So we saw that the PI reporter upon infection, this is 30 seconds after, 13 minutes after updating the bacteria. You can see, I'm going to play it again, if I may. Okay, so the mutant do not branch out, so there's a protrusion, and they have, sorry, I just want to show you better because I feel I'm just, need probably to remove the laser point. Okay, here it is. So yeah, here is a control cell, is stretched, and the PI reporter moves to the membrane and accumulated during the infection. And instead, in the Pexf mutant cell, you see that the reporter is highly concentrated in the middle of the cell, and the cell doesn't stretch. So again, different in the concentration as the lipidomic suggested, difference in distribution as our live imaging suggested. So just to cut a long story short, we perform a genetic interaction of genes that encode for enzyme of this specific branch of the glycerophospholipid pathway. Because apart of the metabolic differences that I showed you in the previous slide, we also found in the transcriptomic analysis that there was a misregulation of the gene encoding for a key enzyme of the main step in the synthesis of PI, starting from that. So we basically overexpress or depleted in the S2 cells each of these enzymes and look at the cytoskeletal protrusion and also at the accumulation of bacteria in the infected sample. And this is one of the key data that we collected. So when we depleted DGK, which basically stopped the synthesis of PA that is very high in the mutant cell, so we saw that if we depleted this enzyme in the Pexf cell, in non-infected cell, you don't really see a particular change in the cytoskeletal protrusion, but when you do it upon infection, you can see that now the cell protrude better than what it was observed in the Pexf alone, although not at the same extent of a control. We observed the same thing if we overexpress CDS1. We also found a partial rescue of this cytoskeletal protrusion. So we wanted to ask, what does this mean in terms of survival to an infection in a live animal? So again, we use the same system I used during my postdoc, the GAL4US system. We have intrasoftware system we borrow from the yeast, where we have a strain that expresses the transcription factor GAL4 under a tissue or a temporal specific promoter. And we cross these two flies that we can buy from stock center, where under the enhancer UAS sequence target of GAL4, we express inverted repeat for a gene of interest, so that in the F1, the inverted repeat produce a double-strand RNA, which then produce siRNA, and the target the transcript of your gene of interest, and then you have a depletion of this gene. So in this particular slide, I was depleting in the macrophages Pexf, so to have fly with no functional peroxisome in the macrophages. So when we saw that, once again, when we did that, then we injected the fly with a bacterium and the ECC15, and then we saw again, first of all, that the Pex knockdown fly, they die faster than the relative control, that is in gray here. And then when we overexpressed the DGK enzyme along and in the Pexf control, and what we found that by overexpressing DGK, which increases the production of PA, we can have higher mortality compared to the Pexf alone, suggesting that by disturbing this metabolic pathway and shifting the pathway towards the production of PA precursor can affect the ability of the animal to clean the infection, and specifically in macrophages. And then we did the other experiment, the complementary experiment by manipulating CDS1, also known as CDSA. So CDS1, the knockdown can accumulate also PA in the cell. And so we have flies with the knockdown CDSA macrophages and in macrophages. And then we found that, again, while Pexf die faster than the relative control upon infection with DCC15, when we knock down CDSA alone, so we accumulated PA, we saw that the death rate is similar to the Pexf RNAi alone. And when we do this, but if we ever express a CDSA, so we reduce the PA concentration in the macrophage of this fly, we found that the ability to survive to infection now is improved and closer to the control level. So suggesting that this lipid pathway affected by manipulating the cytoskeleton of the cell affected the ability of the cell to actually recognize and fight the infection, and therefore improve the survival of the cell, of the animal. Oops. So then the next question was like about how molecularly this accumulation of different lipids is affecting the ability of the cell to stretch the cytoskeleton and to gain some more functional cell. And so one thing that is very well known and is also well known in the clinical world is that the cytoskeleton remodeling, and mainly the raw GTPase-mediated cytoskeleton remodeling, are dependent on the accumulation in the lipid raft of these glycerides and glycerophospholipids like PI and the derived PIPs, DAG, and PA. And we know also raw GTPase can impair immunocell activity. In fact, they are the most mutated genes in many immunodeficiencies. And so we went to see whether, in our case, the signaling affected was the raw GTPase signaling. So what we did, again, we have our control, Pexf mutant macrophages, and then we have the raw cell that overexpress a raw dominant negative. And we found that when these cells were infected while control stretched the cytoskeleton, the Pex1 and the raw NA19 cannot. However, when we express the Rho1V14, which is a constitutively active form of Rho1, we have now a stretched cytoskeleton, the Rho1V14 alone, and the Pex1 now can rescue the membrane raft upon infection. So we suspected that maybe this change in lipids can affect this signaling. And we wanted to test again this in vivo. And this time we use a more aggressive pathogen for the fly, that is Pseudomonas entomophila. And so we tested the raw constitutively active flies, the Pex5 knockdown, and the Pex5 where we overexpress raw. And what we saw again is the Pex5 alone, they are very sensitive to the PA infection. And then when we overexpress raw in this flight, now we see that the level of survival are closer to the control survival. So by activating this pathway, we can rescue the activity of macrophages and the survival of the animal to infection. But how is this happening? We wanted to gain a little bit more insight into the mechanism. So we went back to our transcriptomic screen and we analyzed the hit by GCA, enrichment analysis, and cytoscape analysis. And we found that when we were comparing control versus Pex5 infected gene change to the control versus Pex5 knockdown mock infected, there were differences in the pool of genes that were deregulated, and in the category that fell under the name of cortical actin cytoskeleton organization, regulator, actin filament reorganization. So we went to look at the list of these genes, and we found that 5 genes belonging to the family of raw gap, they were particularly upregulated. And what are a raw gap? So the raw GTPase, they all work in this way. They are normally in the cytoplasm in an inactive form, bound to a GDP molecule. Upon signal, when they have to get activated, the GDP group is exchanged with the GDP by the GEF protein, so that the signal and effector molecules are activated. However, when the signal needs to be turned off, a protein called GAP, which is what is upregulated in my screen, they remove the GTP and add the GDP. So by turning off the signal. So we perform a mini screen to see whether this downregulation of this GTP in Pex1 was changing the cytoskeletal phenotype. And of all these GTP, we found that raw GAP P190, which is a conserved GAP protein from Drosophila to humans, when it was knocked down in Pex1 cell, allow the cell to rescue their ability to stretch the cytoskeleton during infection. And when we overexpressed the raw GAP 190 in control cell under an active promoter, we found that the wild-type cell lose their ability to stretch, suggesting that this signaling is turning off the raw and therefore is impairing macrophages activity. And finally, that to understand whether this was connected with the metabolic dysfunction we had observed before, we went back to the cell line system and we tracked the Rho gap and phosphatidic acid in the cell. And we found that were in control cell infected, the phosphatidic acid is central, accumulated on the nuclear membrane as reported in literature. Rho gap is in the cytoplasm. In the Pexf1, we found that Rho gap is at the membrane and colocalized with the PA reporter. However, when we knock down DGK, which, as I told you, is able to reduce the PA in the cell, we found that now the Rho gap 190 is in the cytoplasm, and the PA also is reduced. So in this way, we connected peroxisome to the production of these lipids that are important to signal the Rho GTPase, activate the macrophages to clean up the infection. However, this signal, as I told you, is a very core signal in macrophages and immunity because they have been linked to migration of the cell to site of infection, development of the cell and differentiation, and also pro-inflammatory phenotype. And that's why their depletion is linked to immunodeficiency, because the cell have no way to activate their inflammatory phenotype. So because of Drosophila, we tested the phagocytosis and we found that the phagocytosis defects are also linked to this signal. But we also know that Drosophila macrophages during an infection, like the mammalian macrophages, they release inflammatory mediator like interleukin-6, which in Drosophila is called unpaired 3. So we want to see in vivo whether affecting these, having defect in the Rho signaling because of lack of peroxisome can also affect secretion of inflammatory mediators. And so we saw that a fly that expressed unpaired 3 in the macrophages, upon infection with DCC15 at systemic level, they secrete in the abdominal cavity unpaired 3. But fly that do not have functional peroxisome cannot. And then when we overexpress Rho V14, the active 4, we can rescue the ability of this cell to secrete unpaired 3. And in the same way, oh, sorry, it's down here. And that likewise, Rho V14 alone, it's not going to affect this secretion. So from this work, we concluded that peroxisome are important for different activity of the macrophages because they control this core signaling pathway through release of important lipid mediators. Now, what we like to do for having an immunological relevance is like to see what's happening in fly and test if the signal is conserved in mammals. And we always like to start with the murine system. So for this experiment, I'm going to show you what's happening in macrophages, bone marrow-derived macrophages extracted from a mouse that is mutated for Pexf2. And we changed the mouse just because this is viable and the Pexf5, it's not, or the Pexf1. So what we found is we went straight to test for the secretion of inflammatory cytokine. So we performed immunofluorescence against IL-6 and TNF-alpha on wild type and Pexf2 homozygous mutant macrophages. And we found that we can detect TNF and IL-6 in the cell of both genotypes. However, when we went to measure by ELISA their secretion, we found that in the Pexf2 homozygous, we cannot detect the secretion of none of these cytokines, suggesting a similar phenotype observed in the fly. So we performed a different experiment, just showing you here key one. And the first one was like to see, is the phosphatidic acid responsible for that? And so we treated the cell, the Pexf2 and the wild type cell, and with the free fatty acid, which are also accumulated in cell with no functional peroxisome, specifically phosphatidic acid, and then with the niacin, which is a scavenger for fatty acid accumulation. And what we found is the free fatty acid and also phosphatidic acid inhibiting the ability of these macrophages upon LPS stimulation to secrete inflammatory cytokine. However, when we treated the Pexf2 mutant with a scavenger for free fatty acid, we can rescue the ability of this cell to secrete interleukin-6. So we demonstrate in this way that the imbalance in the specific lipids in mammalian macrophages also affect cytokine secretion. So the next question was, and is this Rho GTPase dependent? And so to demonstrate that, we treated the cell with two independent Rho-GTPase inhibitor, rosin and HOP16. And we found a wild-type cell treated with rosin or with the other inhibitor lose their ability to secrete interleukin-6, suggesting so that the pathway is conserved in mammals. And we also checked to see if this membrane environment is important just for signals that are activated by membrane toll receptor, because LPS activate TLR4, which is a membrane toll, or from intracellular toll. So we tested the antagonist for other toll receptor except the LPS. So we tested PAM3CSK4, which activated TLR1 and TLR2 membrane toll, and then we used the R848, which activated toll 7 and 8, which are intracellular. And what we found is that when we activated the cell with the TLR, with the PAM3CSK4, the Pexf cell cannot secrete interleukin-6. However, when we activate it with intracellular toll, they can, suggesting even more that the change in lipid milieu at the plasma membrane is what affects the signaling that promote the macrophage activation. We demonstrated also in that this signal, the dependence of peroxisome and peroxisome lipid weights are important not only in macrophages, but we tested from the same mouse, mast cell, and we demonstrated, I'm just showing you here a graphic abstract, the wild-type cell need a peroxisome to secrete inflammatory cytokine upon IgE activation, but not for the granules release. And in fact, the Pexf cell, upon IgE activation, they show a very low secretion of inflammatory cytokines. So then, we work in a clinical department, so we like to collaborate with our colleague and see if our discovery can have an implication to human health. And so I collaborated with Dr. Beata Di Falvi, which is a pediatric immunologist. She did not have at the time any sample from immunodeficiency patient, but she did have a sample collected from her patient with a high inflammatory disease, such as juvenile arthritis and Kawasaki disease. Both diseases are characterized from a high amount of inflammatory cytokine. So at that point, we wanted to see, one, are peroxisome supporting or increasing in this inflammatory environment, and two, are peroxisome metabolites we identified in fly and mouse important in these immunocells. So just I'm showing you just one of the results. So we found that in Kawasaki disease, PBMC, peroxisome number is way higher, as demonstrated by SIKL staining, than in healthy control PBMC. And we also measured the phosphatidic acid because we assumed that if a high level of phosphatidic acid inhibit the cytokine secretion, cells that are secreting a lot of cytokine, they might have lower phosphatidic acid. So we performed a biochemical assay and we did find a reduction of phosphatidic acid in the membrane of Kawasaki disease PBMC. So, but from this study, we basically added up to the list of diseases that have immunodisease or immune-related diseases that have seen a correlation between peroxisome function and immunophenotype. In fact, like there are other disorders that, like that leukopathie, that they have a very activated inflammatory microglia that their phenotype depend by dysfunction peroxisome, specific peroxisome beta oxidation. And we also have evidence that Alzheimer's, Parkinson's inflammatory phenotype depend on peroxisome activity, or peroxisome is not exclusively involved, but is part of the players. So one question I always get when I try to build up the case that peroxisome can be an important marker or target in immunodisease, it's always like, but how about the peroxisome biogenesis disorder patients? Because Peroxisome are well known because their constitutive depletion caused a developmental disease that they fell under the umbrella of Zellweger syndrome, which is a very severe neurodegenerative and neurodevelopmental disorder, and that mainly affects children. And it is a rare disease. So in collaboration with a doctor, which is a pediatric geneticist that take care of a lot of PBD patients, in the number that we can say for a rare disease, she supplied data from the NIH of 500 patients of different age and with different mutation. And consistently, we found that the number of neutrophils and lymphocytes in these patients are actually reduced, suggesting that there is an hematopoietic impact caused by the lack of peroxisome. And also some of patients have been reported as case study, especially 3 came out in the last year in Italy, that they show high immunodeficiency phenotype in children with severe Zellweger. So this is what we are working right now. We are about to publish a paper on this. So we wanted to study, okay, so the peroxisome are important in immunocells. Their metabolism is important for the immunocells, but then in a severe case where peroxisome are absent for the entire development in all the tissue, here we have a hint that seems to be saying peroxisome biogenesis disorder kids have an hematopoietic defect. So to gain some insight in that, because this is an aspect of the disease that has never been really considered or analyzed, we used again our mouse model, and we decided to do a drop-seq analysis of the CD45 positive cells, so the immunocells extracted for neonatal livers, and then sequence to identify by single-cell sequencing analysis the immunopopulation in wild type versus the Pexf neonatal liver. And this has been a very challenging experiment because it's very hard to work with these mice. We had to pull a lot of livers, and plus because there is the neonatal liver, D0, has never been really defined, neither by flow, there is just one work about that, and by single cell analysis, even less. So we are we have been working in collaboration with Dr. Hallett at Western University and Tobias Karakash at Dalhousie to try to get the best result. But one thing we can say, we can identify cell population in this neonatal liver, and we have a large amount of granulocytes and B cells. And when we overlap the 2 samples, so the knockout and the wild type, you can see from color-coded analysis here that there is a difference in the immunopopulation of that fell in the same cell cluster. So we are, as I say, these are just, we don't have yet the final graph, and that's what is holding the paper back. But we do have a lot of analysis that allow us to identify different population in both the wild type and the mutant liver, in the neutrophils, in the B cells, and macrophages, and dendritic cell pool. These are a population that have shown the differential gene expression between, as you can see here in red, the wild type, and here in blue, the mutant groups. And so they show some interesting change that tell us 2 things. First, some of these population that we have identified in both liver, they shift in size, and they are basically larger immature population, the wild type, in the Pexf mutant compared to the wild type, but also this heat map that look at 500 genes, the differential gene expression of the 500 genes that change the most between the different phenotype. It point out some interesting data, and between these we found that all the APC cells, so B cell, macrophages, and dendritic cell, they actually show a reduction not only in differentiation, but in some very essential differentiation markers that are the antigen and major histocompatibility factor 2 for antigen presentation to T cell. So The pseudo-analysis was analyzed again for gene enrichment analysis, and we found a large number of genes that fell in the category of negative regulator of cell differentiation. And between these, there was a specific group for dendritic cell differentiation. And in this particular group, we also found that there was a negative regulation of cross-presentation of the soluble antigen. I must say, when we saw this data, they actually were connected to a conversation I had with somebody in Germany, which also had found that peroxisome in macrophage had a downregulation of antigen presentation molecules. So I decided that was an interesting population to study. However, as I mentioned to you, the liver is very hard to work with because it's neonatal, and ethically and experimentally cannot really give a lot of cell for in vitro cell signaling analysis. So we switched to bone marrow derived dendritic cell. And of course, the first thing we had to do when we start to differentiate this cell from the wild-type Pexf mouse was to see, do we still see a downregulation upon differentiation of this molecule? And so flow cytometry analysis showed that in the wild-type population stimulated with LPS, you can see a shift of MHC class 1 and class 2, and also of what are called co-stimulatory molecule important in antigen presentation. And in the knockout, instead, we found is first of all that the MHC class 1 and class 2 are reduced compared to the control, and that stays also upon stimulation with LPS. But then interesting, we found that upon stimulation with LPS, which activated the cell, we have an increase in the expression of the surface of the co-stimulatory factor. That was a very interesting phenotype, because this phenotype was reported to be the same in the dendritic cell from a mouse, a global mutant for CETA. CETA is the transcription factor responsible for all the MHC class 2 molecules. So we want to look at the expression of CETA in the Pexf and the control mouse, and we actually found that upon LPS stimulation, this cell cannot induce CETA as the wild type does. And likewise, the target of CETA is the H2AA chain of the MHC class 2, and while the control can induce upon stimulation this gene, the Pexf dendritic cell cannot, suggesting that there is a problem. Even if you provide exogenous differentiation factor, still the cell cannot, without peroxisome, cannot induce these molecules. And so here we got a little bit of the help from the literature, because it is known that the kinase PKC, specifically the isoform delta, is very important for the activation of CETA transcription and therefore transcription of MHC class 2. These data were reported in macrophages and in dendritic cell. So we were very intrigued by PKC because PKC is a very lipid dependent to be activated. And so we want to look at the PKC activation by using an antibody and immunofluorescent technology for phosphorylated PKC delta, which is a means of activation. And you can see that the wild type with LPS have a higher amount of phosphorylated PKC compared to the Pexf stimulated. But then when we treated both cells with the PMA, which is an analogous of the diacylglycerol, specifically with the medium chain fatty acid, we can activate PKC delta even more in the control stimulated, and also we can rescue the dendritic cell from Pexf. And then we went to also look at the MHC class 2 upon stimulation with PMA in both genotype. And we found again that while MHC class 2 upon stimulation is really high in wild type cell, but not in Pexf dendritic cell, PMA can rescue the expression of the Pexf in the Pexf dendritic cell. So the idea is that as we show in Drosophila, diacylglyceride, glycerophospholipid, they really depend on peroxisome response during stimulation. And potentially, because the DAG is very important in the membrane to recruit a PKC and then promote the cascade that activates CETA, that these could be connected. So to demonstrate that at transcriptional level, we did treatment of wild type and Pexf dendritic cell differentiated in vitro, and then we treated it with the PMA again, which is a strong stimulator, and then we treated it with another analogous of DAG. Is actually called DOG. I didn't write that because it's quite distracting. And we went to look at the transcriptional expression. We found that wild type, either whether you treat with LPS, PMA, or DAG, you do get an increase of expression compared to unstimulated cell. But then in the Pexf, you do get the expression just when you treat with the PMA and DAG. And this is true for CETA. And this is also true, although a less extent, for H2AA. Of course, H2A is a target of CETA, so it could be probably a matter of timing. If we prolong the time, we might see a better induction. Data were also confirmed by Western blot analysis, where we looked at, as we did in the immunofluorescent, to phosphorylated PKC. And we found that the PMA treatment, they can rescue the phosphorylated PKC, although the protein level do not really change. Likewise, PMA can also increase in Pexf the amount of the CETA, which is PKC dependent in the dendritic cell. And finally, we also look at the MHC with DOG, since I show you that DOG can increase the expression of this at transcriptional level. But we also found that immunofluorescence experiment show that the MHC class 2 in wild type and in Pexf mutant can be induced by these analogous of DOG, as well as by PMA. But the implication of having low or high MHC class 2 is even larger in an animal, because the MHC class 2 are essential to activate adaptive immunity. So that was what we took us to look at this phenotype and to study it more in details, because that would mean the peroxisome, although can impact maybe innate immunocell, their implication on the animal immune responses also affected the adaptive immunity. And so again, we did the in vitro experiment, because we cannot work with this animal in vivo, to test the ability of this dendritic cell to present to T cell. And we used two assays. One was the ELISA, so after co-culture of this differentiated stimulated dendritic cell with the CD4 T cell from mouse OT2, which basically the mouse OT2 expressed the T cell receptor specific for ovalbumin, which is a peptide used to stimulate the dendritic cell and make them present to T cell. And so then when these two cells are co-cultured, you can measure whether T cells are activated or not by antigen presentation, either through ELISA and measuring interleukin-2 secretion, or you can measure calcium waves in the T cell itself. So again, we show that the MHC class 2 phenotype, every time we differentiate, we make sure the MHC class 2 phenotype is there in stimulated and non-stimulated, so reduced in the knockout. And then when we tested this cell, we found that a high concentration of albumin, we record in the Pexf mutant less amount of IL-2 secreted compared to the control. Then we repeated the co-culture, this time for doing calcium wave. And so what we do, we get this multi-imaging, and then we analyze with MATLAB. And this experiment is done by Yannick Hamon in Marseille, that is really a kind of a god in this technique. And so you can see, you record the wave in all these imaging over time. And you can see these how these control of not stimulated as a very little increase in calcium, but when you stimulate the control with LPS OVA, you can get a lot of waves. So we did that for different co-culture. And what we found was that the DC that are mutated, they do express they do express like lower sorry, they do not express, they do show lower stimulation of the calcium signaling. However, we also wanted to see if that could have been rescued by the treatment with oh yes, sorry. So we did show this with the lower concentration and a higher concentration just to see how they respond. And we can see here with ovalbumin, the control are always lower in calcium signaling compared to the stimulated wild type. And here we can see with 0.5 micromolar. And then we did do the experiment again, but with this time we used the PMA to try to see whether the PKC-dependent activation can also, of the DC cell, can also improve the antigen presentation. So in this graph that is a little bit more simple, you can see the control is lower, but not at larger sense, very hard to measure in vitro these waves, but is still significantly lower. And then this difference increase when you are stimulated the control and the Pexf. But when you treat with PMA, you can see that now the Pexf dendritic cell are more able to induce a calcium signaling. And again, we checked that the PMA is not affected the dendritic cell differentiation compared to the control. Otherwise, that could be a different mechanism. So in conclusion, we found that the peroxisome metabolism control a different lipid at the membrane, which in different cell line might affect their function and have an implication on the immunity of the animal. And so in this particular case, we look at the ability of the dendritic cell to present to T cell. And so at just to summarize, like now the number of functions which peroxisomes involved in it have been increased by my work and other people work in the last 10 years. And now we know that they are involved in TLR regulation, antigen presentation, phagocytosis, and NF-κB signaling. And therefore, they have to be controlled or maybe targeted when we are looking at different immunodeficiency or inflammatory disease. In fact, as for insulin, we know that there are drugs that can target different enzymes of the glycolysis, for example. It is really inflammatory, and this is the major signal involved in insulin resistance and diabetes. So as the drug have been efficient in targeting different enzymes and ameliorate the condition of the patient, maybe combinatory strategy that involve like affecting, for example, glycolysis or mitochondrial activity and peroxisome could be even more effective. And we have performed a drug screen, and we have a drug that can increase the peroxisome activity, at least in vitro. And that's where we stopped. So we are, the lab is very actively trying to dissect at the moment the role of peroxisome in basic immunosignaling, such as this is a GIZU work and also rehab, but also we are trying to connect this signaling using different models like Drosophila mouse and human to inflammatory diseases such as Stephanie is looking at Alzheimer's and Parkinson, and Marinella is just finishing her work looking at the role in IBD. So with that, sorry if I took a little bit longer, and I would like to thank all the people in the lab that did the work I presented, mainly Anu Natta, she's now gone to dentistry school, past member that participated to the work, and then the current member that are working on this project, and different colleagues that are collaborating in different projects that I described, and the one I could not really